Hi, this is Professor Tiffany Martin with Medical Microbiology, and we are working our way through Chapter 16, Adaptive Immunity. And we've been talking about T cells, but now we need to talk about B cells. Remember, our third line of defense is made up of T lymphocytes and B lymphocytes, so it's time to talk about our B cells. So let's see where we are with that. Okay, so B cells are going to be the ones that are going to produce the antibodies. We'll get to that in a minute. They are made in the red bone marrow. They undergo clonal deletion in the bone marrow. And then once they do that, they're typically migrate into the spleen and the lymph nodes. Some of them may be in the red blood cells. And their major function is going to be secreting antibodies. So that's some information about B cells. They also have a receptor on their surface that's going to give them their specificity, and that is a B cell receptor. So just like the T cell had a T cell receptor, this one has a B cell receptor. Each one is unique between B cells. Each B cell has its own unique B cell receptor, and then that unique receptor is found in multiple copies on that one B cell. We still get that variability. So you remember when we talked about a shirt, a pair of pants, and the shoes, we're getting that same thing to form our B cell receptor. Okay, again, just by statistics, this variability is capable of making enough B cell receptors to recognize millions and millions of different epitopes. So just statistically, we make a B cell that is going to match any bad guy we encounter. Just statistically, because it's so many, it just works out that we will have a B cell that does that. So let's look at that B cell receptor a little more in detail. If we remember the T cell receptor, it had a transmembrane portion. It had some constant regions from that, but it only had one antigen binding site. So contrasting that with the B cell receptor has two antigen binding sites. So when we draw that, a lot of times you'll see them drawn as this Y-shaped, and you know that it's capable of binding two antigens. Now, each of these antigen binding sites are identical. This is a B cell receptor on a B cell. Both of these will bind to that identical soulmate epitope, but it can bind to two of them. So two identical epitopes, and that's going to be important later on. All right, there is a region that's constant. This is called the heavy chain, and we're going to see that B cells are able to produce a couple of different types of heavy chains, and that's going to make a difference in our classes of antibodies that it's capable of making. So we'll keep that in mind as well. All right, so this region is constant. The heavy chain can be swapped out to do a few different types, and then all the variability is up in these antigen binding sites from here. All right, so that's how a B cell receptor looks like. B cells are going to be involved in the humoral immune response. Remember, humor is just an old term for blood and fluids. They recognize exogenous antigens. So our B cells are running around binding to antigens that are present in the body. Not inside a cell like the T cell and the cell-mediated immune response, but just in the body. So they can bind to the bacteria right away, or you remember we've got all these phagocytic cells that are eating bacteria up and then eliminating it, chopping it up into pieces and eliminating it. Some of those pieces will be floating around in our blood and in our lymph, and the B cells can bind to that as well. All right, but each one will only bind to a specific, the soulmate B cell antigen, the antigen that B cell is designed to do. Okay, so we have our B cells running around. We need to know, when do we activate B cells? How do the B cells know when to go? So that's what we're going to talk about now. We're going to talk about T cell dependent antibody immunity and clonal selection. And this is basically B cell activation. All right, so the first thing I want you to do is, this chart should look familiar because you have already watched the video about activation of a cytotoxic T cell. So I want you to stop and look at this, talk your way through it, answer a couple of questions, and review this before we go on. All right, we did that because there's a lot of similarities, and I want to make sure you have T cell activation down before we added B cell activation and how that works, okay? So you remembered you've got your cytotoxic T cells and we've got our helper T cells, and our helper T cells differentiated into T helper 1 cells with the interleukin, and that was the go signal, and we made a whole bunch of clone activated T cells that were headed out to kill cells. Okay, so back to my board. It got a little busier because I've added a few things. Remember, we had our dendritic cell here that started this whole process. Remember, skin and mucous membranes, it found a bad guy. It's presenting it on MHC2, MHC1. We had a cytotoxic T cell with its CD8 bind to MHC1 and identified this antigen that you're designed for is a bad guy. It waited until T helper cell also had a chance to find a soulmate antigen with its CD4 presented on an MHC2. 4 times 2 is 8. So that worked, and it differentiated into T helper 1 cells. It's called T helper 1 because it's making this new interleukin, and that's the go signal. That's what the cytotoxic T cell was waiting on. So then the cytotoxic T cell made an army, and those T cells headed out to go find cells. So I'm going to get rid of my cytotoxic T cell because he's already done its job and has moved out. So meanwhile, we have B cells running through our body. Remember, they were made in the bone marrow. They've got their unique B cell receptors. I'm drawing it in a Y because I've got 2 antigen binding sites. I've already drawn a mistake here. I've got 2 antigen binding sites, and so it's going to bind to antigens from that. It may have found, here I'm going to put 1 here and 1 here because it's binding to those antigen binding sites there. It could have found these soulmate antigens in the tissues or in the lymph system. Remember, B cells are wandering around. It's got these B cell receptors. It has come in contact with pieces of the bad guy. The dendritic cell had some. He had some pieces. We had some pieces there. They were spit out into the, went through exocytosis, and little bitty pieces were in the circulatory system, in the lymphatic system. The B cell found some. So he now has a match, a soulmate for his B cell receptor. But just like a cytotoxic T cell, he is going to wait. He wants a second opinion. He needs a T helper cell to say for sure this is a bad guy. So we need to make sure that we get this signal. So I said that the T helper cell will differentiate into T helper 1, and we know what he did with the cytotoxic T cells. T helper cells will also differentiate into I need a bigger circle, sorry, into T helper 2 cells. And we're going to call them T helper 2 because they make a different interleukin. So they need a new name, so they have a different job. So it started to make a different interleukin. Again, I don't care that you memorize the names of them right now, but this interleukin that he makes is going to be the go signal for my B cell. So T helper 2 cells help B cells from that. Let's go back to the PowerPoint and look through their drawing. Okay, so that was the T cell one that you went through, and we had our T helper cell that differentiated into T helper 1. Now let's look at the B cell. So this is a T cell dependent immune response. So here we go. We have our antigen presenting cell, our dendritic cell. It found its soulmate helper T cell, and then it bound to that. So I have a CD4 bound to my MHC2. So 4 times 2, that's 8. So I know T helper cells bound to that. It found its soulmate antigen, the epitope that's here. There's some more binding that happens, and this causes it to differentiate into a T helper 2 cell. So it helped the T helper 1s, but it's also doing this. So now I have a T helper 2 cell. It is making different interleukins, and it will bind to my B cell. So my T helper 2 cell is going to bind to my B cell here. So I get another complex happening. This is going to activate my B cell. And now my B cell has gotten the go signal. It bound to a T helper 2. It's got that soulmate antigen. It's making signals, interleukin, and it's telling this B cell, I agree. This is a bad guy. You need to go do your job. So what does the B cell do? It differentiates. It's going to change. It's going to start making some new proteins. And now we're going to give it a new name. It's not called a B cell anymore. It's going to be called a plasma cell. And it's going to be able to make antibodies. Now those antibodies, you notice that Y shape? These antibodies are the same thing as this B cell receptor without the transmembrane portion. So instead of the B cell receptor being connected to the B cell with that transmembrane region, these plasma cells can make the same B cell receptor without the transmembrane region, and now we call these antibodies. So a big pet peeve of immunologists is a lot of times people will say B cells make antibodies, and that's not true. Plasma cells their job is to make the antibodies. Now, just like T cells, some of these plasma cells will differentiate into memory B cells, and they will have this transmembrane portion of the B cell receptor on it, and these will remain for decades. And if we ever see this particular bad guy again that started this process, these B cells will go back and differentiate into plasma cells and make more antibodies. This makes that secondary response happen more quickly. Okay. Okay, so just some more words to summarize some things. Plasma cells, that's what the B cells differentiate into after getting their signals after binding to that helper 2 cells. They're going to differentiate into plasma cells, and it's the plasma cells that make the antibodies. They will have molecules that are specific, that antigen binding region, same as the B cell receptors. Plasma cells actually don't live very long. They only work for a few days. They just churn out antibodies, but the antibodies can exist for a long time. Memory B cells, now these do last for a long time. However, they don't make antibodies. They keep their B cells attached with those transmembrane portions. So B cell receptors, it keeps those B cell receptors attached. It has that same epitope again. But they can live a very long time, and if we ever see that antigen again, it does not have to bind to a T helper cell. It can automatically differentiate into a plasma cell and start making antibodies. So, okay, so we will stop there, and then the antibodies in the next video. Again, the B cell, T cell activation and the B cell immune response is some of the biggest concepts in this chapter, so watch this a few times and do some of the activities and other things as well.